Hello, everybody. It gives me great pleasure, and I'm very excited to introduce Dr. Eyal Gottlieb, professor in the Department of Cancer Biology and Vice President of Research, University of Texas MD Anderson Cancer Center at the University of Texas. I'm sorry, I should repeat myself. Eyal received his PhD in molecular cellular biology at the Weizmann Institute of Science in Israel from Moshe Oren's group. He carried out his postdoctoral research in Craig Thompson's laboratory, first at the University of Chicago and then at the University of Pennsylvania. In 2003, he joined the Beatson Institute, Glasgow, as a research group leader in Cancer Research UK. In 2013, Eyal was appointed director of the Cancer Metabolism Research Unit at the Beatson and remained there until 2020. Although after 2016, he maintained two positions in two laboratories for 4 years, one at the Beatson and the other at Technion in Israel Institute of Technology in Haifa as the Laura and Isaac Perlmutter Chair of Cancer Research, moving full-time to Haifa in 2020 as director of the Rappaport Institute for Biomedical Research and Vice Dean for Research, Rappaport Faculty of Medicine. Incidentally, Eyal also holds an MBA in Industrial Energy and Management from Technion, Israel Institute of Technology. Since January 2023, for just 4 months, he has been at the MD Anderson Cancer Center, from where he is zooming in, I presume. I'll just mention a few of his achievements. He has served as editor of journals such as Oncogene and Cell Death and Differentiation, as senior editor for Cancer and Metabolism, and is currently a senior editor for Cancer Research. Eyal has served on several international scientific research and education committees, on expert review panels and advisory boards, and as a consultant for several companies involved in metabolomics and cancer research. Academically, Eyal's contributions to our understanding of cancer metabolism are just too numerous and too diverse to list here. Suffice it to say that over the past 20 years, Dr. Gottlieb has made several paradigm-shifting discoveries related to metabolic adaptations in cancer. His earlier and heavily cited work established a role for metabolic enzymes, succinate dehydrogenase and fumarate hydratase, linking mitochondrial dysfunction with cancer. His group identified a metabolic sensor function for HIF, or hypoxia-inducing factor regulating prolyl hydroxylases. His group discovered a role for the mitochondrial gatekeeper enzyme pyruvate dehydrogenase in oncogene-induced senescence. And over the years, Eyal's group has also generated state-of-the-art metabolomics capabilities to study metabolic transformation in cancer. His lab is currently interested in the role of metabolic reprogramming in cancer and fatty liver disease, combining metabolomics and genetics approaches to identify metabolic pathways that are essential for disease progression, with the aim of developing new treatment strategies. So I believe that is what he will talk about today. So over to you, Dr. Gottlieb.
Thank you so much, Ameeta. What a challenge to repeat all those complicated names of places and etc. Sorry, my convoluted path in the world from Israel to the US to the UK to Israel and back to the US. So, but yes, I'm in Houston now at MD Anderson, and extremely excited to be with you. Unfortunately, not in person, but hopefully another time. And I will indeed, my talk will cover some of the things that Ameeta actually described to you. I'm going to take a little bit of a historic perspective, but most of my talk will discuss unpublished work that we are currently finishing. So let me share the screen. Can you see it okay?
Yes.
All right, good. Now I can't see you, so that's it. It's me and myself. So I should say that while I'm at MD Anderson Cancer Center currently, the work I'm going to describe initiated at the Cancer Research UK Beatson Institute in Glasgow, and followed most of it at the Technion, the Israel Institute of Technology, at the Faculty of Medicine. I'm still building up my lab here at MD Anderson, and the VP research role keeps me busy as well. So, but hopefully in a year time, I'll be able to describe more work that we have done here as well. So the major players in cancer metabolism are excess growth of demand for anabolic demand. Cancer is by definition a process of ever growing or ever proliferating cells that lost the regulatory homeostasis, and therefore they need to build mass, and building mass is a metabolic challenge. Nevertheless, they are still under a metabolic stressful environment. Oxygen limitation, nutrient limitations are key to the metabolic stress to which most solid tumors are exposed to. And finally, there are genetic stressors. Ameeta touched upon them, SDH mutation and fumarate hydratase mutations, succinate dehydrogenase mutations, and there are isocitrate dehydrogenase mutation. And I will touch on all of these issues during my talk. The point is to convert these essentialities into vulnerabilities. And that's what I'm focusing on throughout my career, identifying the changes and trying to develop approaches to target them. One of which is currently in the clinic, which I hope to finish with this talk. A little bit of background for those who forgot the metabolic textbook, and I'm really going to try and keep it simple. But this is an energy metabolic scheme, path from glucose to pyruvate through glycolysis in the cytosol, pyruvate oxidation in the TCA cycle in the mitochondria, the production of reducing equivalents such as NADH and FADH2 that are further oxidized along the electron transport chain, also in the mitochondrial inner membrane, in order to generate the proton gradient that is essential for ATP synthesis by ATP synthase. So this is the catabolic path that leads to energy production. Well, I'm sure we all heard of the Warburg effect, which is, in simple words, is the generation of lactate from pyruvate in the cytosol. Right? This is more or less what the Warburg effect is, except that it's done under aerobic condition. Pasteur discarded this process way before in yeast, where he demonstrated that oxygen limitation would lead to lactate production. And that's the balance between oxidative phosphorylation and glycolysis as different or alternative sources for ATP production. What Otto Warburg claimed is that lactate is produced even when oxygen is available, and his conclusion was that the TCA cycle or electron transport chain or the whole oxidative phosphorylation machinery, which was not very well described at the time, he did contribute to that discovery and received a Nobel for that, working on cytochrome C oxidase. But he claimed that all these processes are probably not functional in cancer, and that leads to lactate production and tumorigenesis. I should say that by and large, this statement was incorrect. And we know very well that most tumors can do oxidative phosphorylation, and some even more than the normal tissues around them, and some are heavily dependent on oxidative phosphorylation as well. So I wouldn't take Warburg as to its word. However, he definitely was the first one to highlight the need for alteration in metabolism, and particularly of glucose metabolism in cells. And funny enough, I work on several types of, very few rare types of cancer in which Warburg was absolutely correct. These are tumors that are derived from cells that are defective in oxidative phosphorylation, particularly due to the loss of tumor suppressors such as succinate dehydrogenase and fumarate hydratase. Now, why would cells produce lactate under anaerobic conditions or any other conditions? The only reason is really electron balance or NADH oxidation, because this process is a reductive process. Pyruvate is reduced to lactate, meaning that electron is added to pyruvate to produce lactate. These electrons derive from NADH in order to make NAD+. And the reason is to manage glycolysis, to keep on running glycolysis effectively in order to generate more ATP. And these are under conditions where NADH cannot be oxidized in the electron transport chain. So this is the sole reason for lactate production for NADH or NAD plus regeneration. Now, this was the catabolic process. However, we cannot dissociate this glycolytic and TCA cycle catabolic processes from anabolic processes. All the non-essential amino acids that our cells can synthesize themselves, if needed, are derived either from glycolytic intermediates or from the TCA cycle. And this is indicated here in brown boxes of the different amino acids. But all the non-essential amino acids are derived, again, from TCA cycle or glycolytic intermediate. Also, glucose contribute to ribose 5-phosphate production, which is the pentose phosphate pathway, as we all know. And ribose 5-phosphate is a key element or key component of nucleotide biosynthetic pathways. But to make nucleotides, you need also the bases, the carbon nitrogen bases, which are by and large derived from glutamine, aspartate, and glycine. So which are non-essential amino acids that themselves, their carbon source can be derived from glucose as well. So you can see the link between catabolism and anabolism, especially for proliferating tissues. Finally, there is the fatty acids and lipid biosynthesis. So fatty acids are derived from cytosolic acetyl-CoA. This is how de novo synthesis of fatty acids is occurring. The cytosolic acetyl-CoA, the majority of it derived from mitochondrial citrate from the TCA cycle. All right, so a process known as citrate excretion to the cytosol and conversion into acetyl-CoA and oxaloacetate by citric acid lyase. So again, the link is obvious. Once fatty acids are synthesized, in order to make phospholipid, glycerol 3-phosphate is required, which derive directly from glycolytic, from glycolysis, from glyceraldehyde 3-phosphate. Again, linking glycolytic energy metabolism to anabolism. One thing that you see here that is extremely important is that the TCA cycle is a cycle. What it means is actually that there is no net gain of carbons in the TCA cycle. Yes, 2 carbons from acetyl group are entering the TCA cycle, converting the 4-carbon oxaloacetate into 6-carbon citrate. However, there are 2 more, they are lost as CO2 during alpha-ketoglutarate dehydrogenase and malate dehydrogenase, and an isocitrate dehydrogenase processes right at the beginning. 2 CO2 molecules are lost. So there is no net gain of carbon during that process. Yet we deplete the TCA cycle in order to make amino acids, nucleotides, and fatty acids. So how is that? How can this be reconciled? For that, what we need is to replenish the TCA cycle, and proliferating cells depends on this replenishment system, so-called anaplerosis. There are 2 major anaplerotic reactions that are documented today. There are several, but these are the main 2. One is glutaminolysis, as it claimed to be essential for tumor growth. And that makes glutamine de facto an essential amino acid that is then catabolized into alpha-ketoglutarate and providing 5 carbons unit to the TCA cycle from glutamine to alpha-ketoglutarate. The other one, which is a more classic anaplerotic reaction, is pyruvate carboxylase that takes the pyruvate and add carbon to it through bicarbonate and make the 4-carbon oxaloacetate, hence donate 4 carbon units to the TCA cycle that are not lost. So pyruvate carboxylase is an essential anaplerotic reaction as well, and keep that in mind. So let's start with genetic stressors, because after everything I told you, one can imagine that the TCA cycle is such an important component in the mitochondrial processes that all the enzymes there would be housekeeping genes. And the definition of a housekeeping gene is a gene that is essential, meaning that if it's lost, the cell is dead. All right, that's what the house, if you don't have housekeeping, the cells collapse. So it was a huge surprise when in 2000 and 2002, 2 enzymes, succinate dehydrogenase and fumarate hydratase, in the TCA cycle were reported to be tumor suppressor genes in genetic predisposition syndromes called hereditary paraganglioma syndrome for SDH and hereditary leiomyomatosis and renal cell cancer for fumarate hydratase. So these are rare syndromes in which in one inherited allele, a mutated loss of function allele of either subunit of succinate dehydrogenase or fumarate hydratase, together with the loss of heterozygosity with a somatic loss of the second allele, will cause cancer. Will cause neuroendocrinic tumors in the case of SDH, mostly paragangliomas and pheochromocytomas, but also contributes to GIST, gastrointestinal stromal tumor, and to other types of cancer, including colon cancer. Fumarate hydratase loss would contribute to some benign form of smooth muscle growth, some smooth muscle myosarcomas, malignant, but mostly and more relevant to this talk, to the most aggressive form of renal cell cancer. It's an invasive and metastatic renal cell cancer that is associated with fumarate hydratase loss. So these discoveries were made in 2000 and 2002 overall. Later on, after the Human Genome Project was finalized and the Cancer Genome Atlas was initiated, one of the first fruit of those studies in cancer mutations was the discovery of isocitrate dehydrogenase mutations in cancer. Now, these were not a loss of function mutation, but rather a gain of function mutation, or actually generating a neuroenzyme that produces now a new metabolites. I'll touch upon it in a second. These mutations are not also germline mutation. They are mostly somatic mutation. They are typical oncogenic events, and they lead to low-grade glioma by and large, and in about 10 to 20% of acute myelogenous leukemia as well are associated with isocitrate dehydrogenase mutations. So what's the mechanism? Why loss of, I'm going to focus on the loss of function of SDH and FH. Why loss of such housekeeping genes, it's supposed to kill the cell, actually do exactly the opposite and convert them into tumor cells? This was the question I first took when I started my own lab in the early 2000s. So what we know and what we knew at the time is that loss of SDH, for instance, will lead, as I said, paraganglioma, pheochromocytomas, and renal cell cancer. And we knew that these tumors were hypervascularized due to high VEGF and high HIF level inside the tumor. Now, obviously, one could not ignore the overlap or somewhat overlap between VHL, another known syndrome, von Hippel-Lindau, tumor suppressor gene loss, that also cause renal cell cancer, paragangliomas, and pheochromocytomas. And VHL is a ubiquitin ligase of HIF, as we studied very nicely, as we learned very nicely from Bill Kaelin, for instance, and Peter Ratcliffe, who received a Nobel for that. So what might be the link between those components? How SDH can play between VHL and HIF regulation? Or does it affect oxygen sensing? That was the question I asked. Bear in mind that it resides in the mitochondria, where oxygen is mostly utilized for energy purposes. So the work that was done and published in 2005, 2007 by two amazing women in my lab, Mary Selak and Elaine McKenzie, which sadly both of them passed away in 2021 due to cancer within 6 months from each other. So I'd like to remember them with that. So what did they show, Mary and Elaine? So this is the mechanism by which oxygen is sensed. Molecular oxygen is split into two atoms by prolyl hydroxylases. I'll show it again. And that hydroxylate HIF on a proline residue, specific proline residue, which then the hydroxylated proline is a docking site for VHL that target HIF for degradation. That's the way oxygen is sensed. VHL ubiquitinate HIF, and HIF is degraded. However, this is only one atom of oxygen here of the two of molecular oxygen. The other one is used to oxidize alpha-ketoglutarate into succinate. And this is the oxidative decarboxylation of alpha-ketoglutarate in order to prevent production of free radicals. And now you might start seeing the link. Succinate here is a product of that enzyme, the hydroxylases, and SDH, succinate is the substrate for succinate dehydrogenase in the mitochondria. So we tested the hypothesis whether the loss of succinate dehydrogenase will accumulate succinate and inhibit prolyl hydroxylase, hence leading to HIF accumulation in the presence of oxygen and in the presence of VHL. That's exactly what Mary and Elaine showed. That's the first time we started to use mass spectrometry, and that was the early days. Metabolomics in cancer was not even considered at the time. So this is where introduced some work. This is a gas chromatography retention time plus the mass and specific mass that we look for, which is in this case succinate and deuterated succinate that is served as a reference. And then we knocked down succinate dehydrogenase subunit D, and we could see the accumulation of succinate in the cells. And we can quantify it and then test the hypothesis whether succinate can indeed inhibit hydroxylation. So this is an in vitro hydroxylation assay using a HIF peptide that when hydroxylated, it moves faster on SDS page gel electrophoresis. And so this is the hydroxylation reaction, and with increasing amount of succinate, we can see the retention of that peptide polypeptide on the gel, meaning the inhibition of the hydroxylation process. Interestingly, this is a competitive inhibition. So when we increase the concentration of alpha-ketoglutarate in this reaction, maintaining high levels of succinate, we could reverse the inhibition. So that's a classic competition between succinate and alpha-ketoglutarate, which we know very well now they compete on the same binding site for alpha-ketoglutarate. But that suggested to us that maybe we cannot fix succinate dehydrogenase loss, but we can increase alpha-ketoglutarate level to overcome the effect of succinate. And that was the idea, to increase alpha-ketoglutarate and reactivate prolyl hydroxylase and retarget HIF for degradation. So for that, we had to synthesize cell-permeable alpha-ketoglutarate esters and mono and diesters. Some of them are indicated here. And this would get into cells effectively deesterified and increase the intracellular levels of alpha-ketoglutarate. And indeed, we could restore low levels of HIF in the absence of functional succinate dehydrogenase. So really, we confirm the theory that this can be regulated by alpha-ketoglutarate and succinate. We tried to commercialize it, admittedly, unsuccessfully, and probably for good reason, because it's very difficult to use a metabolite as a drug where you need to continuously provide it. And also considering that at the end of the day, when you utilize it, you make succinate, which is the problem of those tumors. So, but that was the early days and maybe naive days that I started to think about utilizing our knowledge into therapeutics. So later on, genetics came to our help in validating this biochemical study that we have done. This is an oncoplot for paragangliomas patients. Every column is a patient, and every row is a specific alteration, oncogenic alteration. Some are germline, some somatic, and some are copy number associated. So what we learned from that oncoplot, we learned that succinate dehydrogenase subunit B is in germline mutation is seen in about 9% of patients with loss of heterozygosity in the tumors. Now we knew that before, but anyway, that's very nicely confirmed here. The same is true for SDHD as well, subunit D in paragangliomas. VHL as well, yes, I told you already, is known to be a tumor suppressor associated with those type of neuroendocrine tumors. So you can see that too. So far, I didn't tell you anything new. I knew that already before we even started our biochemical studies. The interesting start came when, at least in one family, germline mutation in EGottleib was identified to be a tumor suppressor in generating neuroendocrine tumors. EGottleib is prolyl hydroxylase 2. That's the gene encoding for the oxygen sensor that hydroxylates HIF. And this mutation also lead to loss of function and loss of hydroxylation HIF1 alpha. Even more reassuring were actually the discovery that in 5% of neuroendocrine paragangliomas, EPAS1 is mutated, and this is an oncogenic mutation on a gene known as EPAS1. Those who are not familiar with that gene, this is HIF2 alpha. Okay, and the mutation is the proline residue that gets hydroxylated. Okay, so you lose that ability to hydroxylate HIF, and that is an oncogenic event in paragangliomas. And also bear in mind that these are all mutually exclusive, that they all probably fits in the same pathway, so there is no selection advantage once you acquire one type of mutations. You don't get the other. Okay, so putting them together as potential epistatic genetically, and definitely those that are affecting the exact same pathway. So that is very relevant now because belzutifan, which is a HIF2 alpha inhibitor, was recently approved for renal cell cancer with VHL mutations. And nowadays, we are, as well at MD Anderson and other places, we're going to test whether belzutifan is going to be effective in tumors where SDH is lost. And for me, it's a complete full cycle, almost 20 years since we made the discovery that the succinate dehydrogenase loss leads to HIF accumulation, and it's part of the driver mechanism to tumorigenesis. The world of oncometabolism has grown exponentially since those early days, and now we know that beyond the effect on HIF prolyl hydroxylase, there are several other dioxygenases, as they call the split molecular oxygen, that beyond HIF hydroxylases. Many of them are associated with the epigenome. They are demethylases. Both histone and cytosine demethylases are also alpha ketoglutarate and oxygen-dependent dioxygenases that make an hydroxymethyl group that is then removed as methanol as part of the demethylation process, while at the same time oxidizing alpha ketoglutarate to succinate. And all of these enzymes are some more, some less inhibited by succinate and also by fumarate. In the case of fumarate hydratase loss, in a very similar mechanism, which I didn't touch upon, but basically it's repeating what we demonstrated for succinate as well. And in the case of isocitrate dehydrogenase oncogenic mutations, the production of 2-hydroxyglutarate, which is the product of this a new enzyme that is formed due to the oncogenic mutations of isocitrate dehydrogenase that now work reductively and reduce alpha ketoglutarate to 2-hydroxyglutarate, and that's also a competitive inhibitor of dioxygenases, particularly the demethylases. So now I want to focus a little bit more on fumarate hydratase loss of function, and how we're going to capitalize on knowledge and research in metabolism in order to try and address clinical needs for those tumors. Okay, so loss of fumarate hydratase is associated, as I said, with the most aggressive form of renal cell cancer. It's involved in other pathologies I'm not going to touch upon, but also a lower level of fumarate hydratase is associated with poorer prognosis even in other types of cancer, renal cancer, such as clear cell renal cell carcinoma. This is the metastatic view of HLRCC, hereditary lyomatosis renal cell cancer patient, and maybe more importantly, it is essential to discover, because of the invasive and metastatic state of this disease, it is extremely important to detect those diseases at early stages. You can see stage 1 and 2 are potentially curable, while stage 3 and 4 are very little form of renal cancer. There's a huge dramatic difference. The problem is that most, if not all of those patients, as you can see by the number of cases per each line, are discovered at a later stage and usually already at the metastatic, because these tumors are metastasized at a very early stage. So once a patient is admitted to hospital and RCC with FH deficiency is discovered, it is most likely already metastasized to the spine. So there is a very urgent need for early detection of this disease. Furthermore, the prevalence of FH mutations in the population is 1 in 1,000. All right, which is not that rare. However, the penetrance is pretty low. It's 5 to 10%. But nevertheless, there is a high population number of people at risk for developing the most aggressive form of renal cell cancer, even though it is a rare, leading to about 16,000 cases per year in the US. Sorry, 16,000 altogether, 500 new cases every year. So it's a very rare event from a cancer perspective. Okay, so how do we approach that? So first we made the genetic model, mouse genetic models, where we fluxed the fumarate hydratase allele, as you can see here. The study was initiated with ex vivo renal cells that were transformed in vitro, and then fumarate hydratase was knocked out with adeno-3 expression, and we could lose the fumarate hydratase. We can increase fumarate, decrease malate by 100-fold and 10-fold respectively. A clear block of fumarate hydratase activity. This work was done by Christian Fretzer in the lab at the time in Glasgow. Now Christian's a professor at Köln in Germany. This work led to, by multiple publications, is a map, a metabolic map of what is specifically unique about fumarate hydratase deficient tumors. And I'll cover it very quickly. This is about 10 years of work. Okay, so first, we discovered that fumarate is highly present in these cells, as I show you. The cells are completely Warburgian, meaning that they take a lot of glucose and produce a lot of lactate, as one would expect. First discovery that was unexpected is the high production of bilirubin due to increased levels of heme oxygenase and heme catabolism. Now, this process is tackling redox stress in particular, and heme oxygenase becomes synthetic lethal with fumarate hydratase, meaning that it becomes essential for survival of cells that lost fumarate hydratase. Then we looked at the urea cycle as well. We showed high level of argininosuccinate secretion in the cells. And interestingly, that was because of fumarate pushing back a reaction of argininosuccinate lyase that typically take argininosuccinate and convert it into fumarate and arginine. And now high levels of fumarate in the cells push this reaction back and make the cells dependent on arginine. So arginine in the circulation become very essential for fumarate hydratase deficient renal cell cancer. And that is also clinically relevant because only at the AACR, last AACR meeting, there was a description of the phase 3 clinical trial with an enzymatic treatment for arginine depletion in cells that are deficient, in cancer cells that are deficient for argininosuccinate synthase, one step above argininosuccinate lyase. But nevertheless, this approach, depleting arginine, we demonstrated it to be essential for fumarate hydratase deficient cells. So again, there is a possibility to utilizing this, hopefully soon to be approved therapy in FH deficient tumors. Finally, we discovered a kind of an interesting molecule called succinic glutathione. This is a molecule that is derived from fumarate succination, meaning that the double bond here on fumarate is very reactive, an electrophilic attack that actually provide or alkylate the sulfur and make an SC bond, losing the double bond here and make a succinate attach in a thioether to cysteines. In this case, this is the cysteine on glutathione, which is massively reduced in the presence of fumarate in the cytosol, and that leads to a redox stress that is mediated partly through heme oxygenase, as I mentioned earlier. Okay, so this is a reaction that is well described in the world. It's not an enzymatic reaction; it's called Michael addition. And we know that fumarate can succinate thiols in general. What is unique about this study is that happens specifically on glutathione and leads to glutathione depletion in cells. But the succination of proteins, the cysteine on protein, was characterized before, particularly in diabetes. But now, following our and other people's discovery, it was shown that protein succination can also occur in fumarate hydratase deficient cells. And nowadays, the antibodies against succinic cysteines on protein is used as a diagnostic tool together with fumarate hydratase immunohistochemistry, where FH negative, 2SC positive are considered to be renal cell cancer that are FH deficient. And as I mentioned, they are highly aggressive tumors. So this has now become an important biomarker for clinical treatments. However, it requires biopsies. And usually we cannot take biopsies from these patients. The first time you actually see material is after resection of the tumor with surgery. And this is what is done. When there is a suspicion of FH mutation, there is an aggressive surgery, and then you can confirm that it is an FH mutant. But then it's too late, as I indicated earlier, that we need much earlier stages of detection of the disease. Therefore, we were looking for circulating metabolites. Now, in our study from cells, we demonstrated that there is another molecule that is secreted from the cells, and that's succinic cysteine. It's just cysteine that is succinated. And we assume that it's derived from the high level of succinic glutathione inside the cells. To confirm that, we injected succinic glutathione to mice, and then very quickly we saw an increase in succinic cysteines within minutes in the circulation, and then it was clear. However, when we did that in blood samples not in vivo, but ex vivo, then we couldn't see that effect. So we assumed that there is a need also for tissues. It doesn't happen clearly in the circulation only. To cut a long story short, we show that kidney extract specifically can deplete succinic glutathione in an in vitro reaction, generate at first step a metabolite called succinic cysteine glycine. This is a removal of the glutamyl, gamma glutamyl part from glutathione. This is what left behind. And then in a second reaction, it will remove the glycine as well and generate succinic cysteine that is accumulated and is stabilized in this reaction. So succinic cysteine may become an important biomarker for FH detection. So we, together with Leon Zhang and a group of clinicians in Shanghai Children's Hospital, we collected a cohort of 70 individuals with fumarate hydratase mutations and germline mutations and somatic mutations with renal cell cancer. And we first started a pilot study with 10 patients from each group: FH deficient renal cell cancer, FH wild type renal cell cancer, and normal control. And as you can see, when we applied metabolomics, large scale metabolomics and lipidomics study in the plasma sample from this group, and you can see the separation of the FH deficient cells. Zooming in, we could identify a very good classifier, particularly in the red line, as you can see here in this rock curve, is succinic cysteine, which is highly present in FH deficient patients. And also succinyl adenosine, which is another molecule that we have discovered. I will not touch upon it due to lack of time. We know exactly how these metabolites are being produced. We did a mouse model for it with a PDX in this case, where we took one PDX from FH deficient and one from FH wild type patients, and they grew more or less to the same level. This is the confirmation, the current confirmation that this is an FH deficient, again, based on succinic cysteine positivity in immunohistochemistry and FH negativity. They grew more or less at the same rate, but more importantly, succinic cysteine was discovered at significantly high level even at 7 days after transplantation, very early stage, and obviously went up through the roof later on when tumor progressed. When tumor were resected, removed from these mice, the succinic cysteine level dropped down to baseline level and retained low when there was no tumor. So that makes succinic cysteine an excellent potential biomarker. We went back to the whole cohort. Now we studied about 70 to 80 individuals. Is there a question there, or someone needs to be muted? Oh, sorry. Ameeta, I'll be happy if someone.
Please mute yourself.
Okay, thank you.
Okay, I think I got it.
Yeah, thanks. I hope I'm not muting now. No, I'm good. Okay. All right, now we move back to the whole cohort of patients, and you can see how they separated very clearly on the succinic cysteine level in the population, and the area under the curve now cover almost 100%, 98% of the patient with specificity and sensitivity. And we quantify also the succinic cysteine. We made a cutoff with a positive predictive power of above 95%. So we can clearly demonstrate that succinic cysteine is ready to go to the clinic biomarker for early detection. It's also highly correlative with tumor volume. So we could see that as opposed to other metabolites that we discovered in this study, and it's not induced in the carrier. So the heterozygous carrier have similar levels to the normal control tumors. Okay. More importantly, we could see that even at stage 1 and 2 patients, we can already very, very nicely detect high levels of succinic cysteine in those patients. So that raised the question now, can we indeed reverse that curve? And now with the frequent monitoring of a simple metabolite, can actually push our clinical stage to a level where we can intervene clinically at the stage 1 and 2, at stage 1 and 2 rather than stage 3 and 4. That will make a huge impact on lives of these patients. Also, we can use this molecule to monitor longitudinally some patients. This is just an example of what happened after partial resection. This is pre-resection, post-partial resection, after complete nephrectomy, and you can see it here. Then recurrent disease, another surgery, recurrent disease, and then on treatment. Okay, so clearly we can monitor. We can monitor the disease along the process of each individual patient. I'll switch gear and move to therapeutic intervention. And this started from succinate dehydrogenase deficient tumors. But whatever I say here is completely relevant to fumarate hydratase deficient tumors as well. And we have tested it in both systems. So this is the same approach we took for FH. We made SDH deficient mouse model with SDHB flux allele, ex vivo analysis, knockout of SDHB. That's a work done by Simone Cardacci in the lab. And again, it was published a while ago. So to cut a long story short, what we discover is pyruvate carboxylase, which I spent some time introducing you to earlier. This is the anaplerotic reaction that convert pyruvate to oxaloacetate, becomes an essential component in SDH deficient tumors. And the reason is because this is becoming the only way to produce oxaloacetate, which is required for producing aspartate. Aspartate is a non-essential amino acid, but unlike many other non-essential amino acids, most of aspartate is derived from de novo synthesis rather than a mix between circulating aspartate and synthesis. That's because the aspartate level in our circulation is very low to barely detected, and there are no official effective specialized aspartate transporters on most epithelial cells, except for the liver. And then aspartate is an essential component in nucleotide biosynthesis, as I also indicated earlier, and it requires for pyrimidine biosynthesis. And it becomes a limiting factor in SDH deficient cells, hence the production of aspartate from pyruvate and from oxaloacetate, which require pyruvate carboxylase, is essential. You can see it here with the synthetic lethality, in vivo synthetic lethality. This is tumor growth of wild-type cells with and without knockdown for pyruvate carboxylase. While knockout cells can grow, they grow slower in our model as a tumor, but they are completely dependent on pyruvate carboxylase. When we knock pyruvate carboxylase down, these cells do not grow. And those that do grow reverse and overcome the knockdown of pyruvate carboxylase. So PC and SDH and FH as well are synthetic lethal. So we thought, okay, maybe we can target pyruvate carboxylase. At that time, I already started a company that looked for drug, small molecule generation for some metabolic targets. Pyruvate carboxylase was one of them, but it ended up being a very tricky target for reasons I'm not going to talk about. But what we have done is we utilized the cell lines that we generated, the wild type and SDH wild type and knockout and FH wild type and knockout isogenic cells, in order to do a so-called phenotypic-based drug discovery, meaning that instead of searching for molecules that inhibit a specific enzyme, we did a chemical library of drug-like molecules on cells, and we look for the overall effect on the viability of these cells, searching for those that are affecting the viability of knockout cells, but not of wild-type cells. Okay, so we're looking for a synthetic lethal chemical screen on cells. This is an example of one of our hits, 1383, okay, that demonstrated activity at an EC50 of single unit micromolar in wild-type cells, but sub-micromolar in knockout transformed cells. So there was about a 10-fold window of sensitivity. Then through a process called hit to lead, we took that molecule, modified thousands times, looking for other molecules and areas that can contribute to better efficacy and a better window, a therapeutic window in vitro. And this is what you see now. Now you see a molecule that hardly touches the wild type cells. Now the EC50 is 30, it's higher than the 6 in wild type cells, but in the knockout cells, we improve it by a 10-fold decrease. Now we're talking about 5 nanomolar or 50 nanomolar EC50 in cells, in the knockout cells. So that become our hit, our lead, sorry, 8265. Okay. The only problem when you do phenotypic-based drug discovery is that you don't know what the target is, what the molecular mechanism of this effect. This is where my lab, Orly and Sarah from my lab took that task and tried to address this with a metabolic approach. First, confirming those results, you can see in the IncuSite work how this is translated to growth in real life. Okay, so when we increasing amount of this drug up to 10 micromolar, eventually did not touch the SDH wild type cells, maybe a little bit slow it down, while even at the lowest concentration tested here, the knockout cells could not grow and actually died in the culture. A clear synthetic lethality. But we didn't know what the mechanism. So first thing we did is we treated it for a short time, and then we did RNA-seq to see what is the adaptive mechanism in gene transcription that may indicate to us what is the mechanism of action of this drug. You can see that they are very nicely separated on the PCA study. This was done by Jonathan in the lab, a PhD student. We identify about 30 genes that are significantly altered. They separate the treated and untreated, both in wild type and knockout cells. This is important because we expect the same mechanism to occur in wild type and knockout cells. We don't look for the differences between the wild type and the knockouts. We are looking for the differences between the treated and the untreated. And these are the common ones. Mostly TXNP and ARRDC4, illustrated here, are the most affected genes following treatment, both in wild type and knockout. And they are downregulated. They are basically their expression is blocked immediately after treatment. So what are these two genes actually? These are target of a transcription factor called carbohydrate responsive element binding factor, and this complex, this transcription factor, typically is bound to the mitochondria. It's not getting into the nucleus, and it sends glucose through the phosphorylation of glucose to glucose 6-phosphate, binding to Mondo A and MLX which are then translocate to the nucleus and transcribe the genes, among which is TXNIP and ARDC4. Both of them are regulating as a feedback mechanism to slow down or to prevent glucose uptake into the cells. So there is a rheostat mechanism that too much glucose, high glucose 6-phosphate, shutdown of glucose uptake. Okay, but as you remember, these genes are downregulated, so somehow we block the transcription of those genes. So we suspected that maybe we blocked the glucose 6-phosphate level, okay, in those cells. So we did, and I'll skip that. We did carbon tracing in these cells, and this is C13 labeled glucose, and we've traced it all the way to lactate. And indeed, following the treatment, lactate labeling, lactate production from glucose was down to 0, basically, in the treated cells. This is now minutes after treatment, both in wild type and knockout cells. But that will be also true for glucose 6-phosphate. It goes all the way up to glycolysis. So it does block glucose 6-phosphate levels or production. However, when we looked at glucose labeling, it went up in the cells. So immediately we thought, okay, clear. This is a hexokinase inhibitor. Okay, it's blocking the conversion of glucose to glucose 6-phosphate. We set up a hexokinase activity in purified enzymes, in cellular extract. I'm not going into details due to lack of time. We could not see any effect of the drug on hexokinase. So we thought maybe it's affecting hexokinase localization. Hexokinase is also associated with the mitochondria, and this is where it needs to be functioned, particularly hexokinase 2, the major enzyme in cells, and we could not see any dissociation of hexokinase from the mitochondria. So we had to scratch our head a little bit and think, how do we explain the increase in glucose? So the difference between glucose 6-phosphate to glucose, as you may remember, that glucose is exchanged freely in the cells, and it's captured by hexokinase to be phosphorylated in order to be retained intracellularly. So we thought maybe it's the exchange that is blocked really here. So if we block the transporters, and when we do the extraction of metabolites, we actually wash the cells with PBS prior to extraction. Otherwise, we get massive contamination from metabolites from the medium. During that process, we probably lose glucose to the wash. And when we have the drug, we actually prevent that wash away of glucose. That was our hand-waving explanation. It's quite reasonable to assume that, but we needed to test that. So we used two other inhibitors that are known to inhibit glucose transport. And one of them is massively used. It's not a drug, but it's a good tool compound, cytokeratin B, a pan inhibitor of glucose regulated. And also there was a GLUT1 specific inhibitor developed by Bayer company. And we tested both of them. And indeed, both of them led to the increase in glucose intracellular, as what we call intracellular levels of glucose, and a block in glucose 6-phosphate, similar to the metabolite compound. So we really think that this is the mechanism. So now what we did, we washed the cells in the presence of cytokeratin B. And only the wash itself increased the level of intracellular glucose. So this is the escape. What we see here is the lack of glucose rather than the actual demonstration of what the level of glucose intracellular is. So for those of you who are doing metabolomics and looking for intracellular glucose, bear in mind that what you measure is whatever retained in the cells. When we now block glucose escape with cytokeratin B, we increase it here. However, if we pre-treated the cells with the metabolite compound, we now decrease the intracellular levels of glucose, as one would expect. So in order to confirm that, we look outside of the cells of the glucose uptake rate. What you see here is over time, we incubated cells. This is really for 15 minutes with low amount of glucose, and we look at the outside, see how much of the provided glucose is depleted. This is wild-type cells. Knockout cells deplete more glucose because they are more glycolytic, and that's perfectly fine. And when we treat the cells with either the Bayer compound or with the metabolite compound, we completely block glucose uptake over this time period. So indeed, MTB is a glucose transporter inhibitor. It's not a GLUT1 specific for several reasons. First, we used it on GLUT1 knockout cells and it still blocked glucose uptake. And second, we generated Bay resistance cells that have high level of expression of GLUT3 and GLUT4, and the metabolite compound blocked glucose uptake and killed the SDH deficient Bay-resistant cells as well. So it's a pan-glucose inhibitor. But we needed to confirm that positively. So for that, we used, together with a chemical engineer at the Technion, Avi Schroeder, and his student, Noga, we did vesicles derived from erythrocytes that have level of glucose transporters. These vesicles that are trapping GFP, as you can see here. Now, instead of trapping GFP, we trapped an enzyme called glucose oxidase in the presence of a substrate for it that generate basically during the process hydrogen peroxide, and we put amplex red as sorry, we didn't use a substrate. The substrate is glucose. We did not provide the glucose yet, and we use amplex red as a detector for hydrogen peroxide in those vesicles. And now what you see is that we don't provide glucose. This is the baseline fluorescent. When we provide glucose, there is an increase of that, and when we treat the vesicle with metabolite, we block completely the oxidation of glucose in those vesicles, indicative that this is a glucose transporter inhibitor. The consequences of that is really a massive energy crisis in SDH deficient cells, particularly ATP is crushed, while SDH wild type can survive it by running oxidative phosphorylation effectively. The problem is that if I would have to design a drug against TCA cycle deficient cells, I could imagine that they become dependent on glycolysis or glucose uptake. And I would not dare to go there pharmacologically because of toxic effect. And indeed, we demonstrated in vivo that when we provided this drug, we could see a massive increase in the circulation of glucose level. It's a good biomarker for the activity of this drug, that it blocks glucose uptake in many tissues. And it leads to toxicity, poorly tolerated with 2 mg/kg maximal tolerability. So we do have dose limiting toxicity, mostly due to brain effect. We studied this on GLUT3, and the mice cannot tolerate this reaction. Nevertheless, even when we use the very limited amount that we could provide, we could already see some benefit for SDH deficient tumor growth, as you can see in the green line compared to the blue one. So we switched to the Bay compound, which is specific for GLUT1, which demonstrated by PET imaging that it blocks FDG PET in the tumor. Okay. And then what we could see that SDH deficient cells did not grow very well in the presence of Bay1, at least for 30 days. Okay, so that's a massive effect on tumor growth. Nevertheless, they did pick up very quickly, and I did tell you that it's very easy to generate Bay-resistant cells. And while this is extremely significant, the resistant cells develop very quickly during the process of GLUT1 inhibition. And it is translated to better survival, no doubt, but I think we need to do better. And one of the mechanisms we're looking for is brain-sparing molecule that will retain glucose transporter inhibition, but not in the brain. Okay, at that point, I think I will finish because I ran out of time. And I will skip the last part, which is to do with the anabolic growth, particularly the need. I was very optimistic, as you can see, particularly the need to utilize acetate as a carbon source for tumors. We demonstrated that it is essential for lipid biosynthesis, but also, most importantly, for histone acetylation in the nucleus that are associated with response to DNA damage, so DDR, regulated by acetate utilization to histone acetylation, particularly the H4K16 mark. And therefore, a combination of inhibition of acetate capturing by an enzyme called ACSS2, acetyl-CoA synthase 2, is beneficial for patients. We just finished phase 1 as a single agent, not in combination yet, and hopefully we will enter phase 2 at MD Anderson in the near future with this drug. So that's me. I try to thank the people who did the work along the way. I will quickly highlight again Those I didn't mention, particularly Fat and Bella, there's Bella, who did the metabolomic studies in all cases, and the team from Metabomed who generated the GLUT inhibitor and the ACSS2 inhibitor, Ali Fatai, the CEO, and Andreas Gutierrez as the chief scientific officer of the company and a medicinal chemist himself. So thank you very much. I'll be happy to take questions. I'll stop sharing so I'll see you or you'll see me.
All right. Do we have any questions? Fabulous talk, Eyal. Thank you.
No, thank you. It's a pleasure. I hope I did not overwhelm you with metabolism. I assume you are interested in metabolism to some extent.
Yes. So do we have any questions? I think it's probably 9 o'clock, so we have people moving on. So if there are no questions, I'm going to say thank you to everyone. And thank you for attending. And we had a really good crowd. We had 88 people. That's a large crowd so we had a great seminar. Thank you very much.
Thank you.
All right.
Thank you all.